Hello, I'm Reza from Cyagen. Today, we're going to explore our innovative research into metabolic dysfunction-associated steatohepatitis, known as MASH. We'll discuss how our composite mouse model has been instrumental in the preclinical stages of drug development and the mechanisms underlying this severe liver disease. Metabolic dysfunction-associated steatohepatitis arises from complex interactions between metabolic disorders like obesity and diabetes, leading to fat accumulation in the liver, inflammation, fibrosis, and potentially progressing to cirrhosis or liver cancer. Our goal is to better understand and find effective treatments for this condition. This figure outlines our experimental setup, illustrating the groups of mice used, the diets they were fed, the interventions applied, and the timeline followed for the study. It provides a clear roadmap of our research approach and methodology. We developed a composite model by feeding mice a high-fat diet and administering carbon tetrachloride, which together mimic the liver pathology seen in human MASH. This model helps us study the effects of diet and chemical exposure on liver disease progression. This graph illustrates the significant impact of resmetirom on reducing both body and liver weight in mice subjected to a high-fat diet and carbon tetrachloride. The treated group shows a notable decrease compared to the untreated group, demonstrating the drug's potential to mitigate liver enlargement and overall weight gain associated with MASH. Using resmetirom, we assessed how effectively this drug can reverse or reduce the pathological changes caused by the diet and toxin-induced MASH model. This section discusses changes in liver pathology and serum markers over the treatment period. Detailed bar charts show significant improvements in lipid profiles with resmetirom treatment. Total cholesterol, HDL, and LDL levels were favorably lowered, emphasizing the drug's effectiveness in correcting dyslipidemia, a key factor in MASH progression. This figure shows the positive effects of resmetirom on liver enzymes ALT and AST, which are markers of liver damage. The treated mice displayed substantially lower levels of these enzymes compared to their counterparts, indicating reduced liver injury. We closely examined the liver tissue to evaluate steatosis, inflammation, and fibrosis. The use of resmetirom showed promising results in reducing these pathological features. Here, we present micrographs from H&E staining, oil red O, and PSR staining. Each image demonstrates the reduction in fat deposition, lobular inflammation, and collagen deposition in the liver of treated mice, compared to those on a high-fat diet alone. These images visually confirm the biochemical findings and highlight the therapeutic potential of resmetirom. This graph quantitatively depicts the significant downregulation of mRNA levels of liver fibrosis markers like alpha-SMA and collagen-1 alpha-1. The reductions are statistically significant, underscoring resmetirom's role in hindering the fibrotic process in the liver. At Cyagen, our commitment is to provide advanced research tools like our composite MASH mouse model, which is crucial for the development and testing of new treatments. These models help bridge the gap between laboratory findings and clinical applications, advancing our understanding in treatment of liver diseases. Thank you for joining me to learn about Cyagen's contributions to MASH research. If you're interested in our models or wish to collaborate on MASH research, please don't hesitate to reach out. Stay tuned for more insights from Cyagen, where we continue to advance the frontier of genetic medicine.